Hello everyone. In this class, we'll try to learn about reflexes, their classification and different properties. Reflex is an involuntary, that is automatic response to a stimulus. It depends on integrity of reflex pathway, that is the reflex arc. Here, a sensory impulse is automatically converted into motor effect through the involvement of central nervous system. When a person walks barefooted and he steps over a thorn, he withdraws his affected limb while extends the other limb to support the body weight. The whole action is reflex and does not require presence of will. Reflex arc. It is consist of receptor, an afferent neuron, an efferent neuron, and effector organ. The number of synapses, that is the connection between afferent and efferent neurons, vary from 1 to many hundred. These synapses are located in the brain or spinal cord. The activity in the reflex arc starts in the sensory receptor with receptor potential, which is non-propagated graded response whose magnitude is proportional to the strength of stimulus. This generates all or none action potentials in the neurons. So this is a diagram showing the reflex arc, which consists of receptors, afferent nerve fibers, then interneurons neurons, which acts as integrators. Then we have efferent neurons. Then we have effector and organs. In the afferent nerve, the number of action potential being proportionate to the size of generator potential. In the CNS, the responses are again graded in terms of excitatory postsynaptic potential or inhibitory postsynaptic potential at the synaptic junctions. All or none responses are generated in the efferent nerve. When these reach the effector, they again set up a graded response, that is end plate potential. When the effector is smooth muscle, response submit to produce action potential in the smooth muscle, but when the effector is skeletal, the graded response is always adequate to produce action potential that bring about muscle contraction. So this is showing a reflex arc. Again, the sense organ where they are going to produce action potential. In the afferent neuron, they produce action potential, and again in the synapse they produce EPSP or IPSPs. Again in the efferent neuron there will be generation of the action potential, and at the effector end where there is a neuromuscular junction, they produces end plate potential. Again in the muscle they produce action potential. From above, it is clear that the portions of arc, that is the specialized for transmission, that is they have afferent and different axon muscle membrane. The responses are all or none action potentials. It should be emphasized that the connection between the afferent and efferent neurons usually in the CNS, and activity in the reflex arc is modified by multiple inputs converging on the efferent neurons. Note that the afferent neurons enter via dorsal roots or cranial nerves. The efferent nerves leave via ventral root or corresponding motor cranial nerves. The principle that in the spinal cord the dorsal roots are sensory and the ventral roots are motor and is known as Bell-Megendie law. Coming to the classification of reflexes, reflexes can be classified as a monosynaptic reflex, polysynaptic reflexes. Monosynaptic reflexes which only one synapse is present between the afferent and efferent neurons. For example, all stretch reflexes, biceps, triceps, and knee jerk. Polysynaptic reflexes, the reflexes in which one or more interneurons are present between the afferent and efferent neurons. For example, withdrawal reflexes, flexor reflexes, cross extensor reflexes. Coming to monosynaptic reflexes, which are called as stretch reflexes or myotatic reflexes, when skeletal muscle with an intact nerve supply is stretched, it contracts, which is called as stretch reflex. And this response, it is a stretch reflex. Here, the stimulus is stretched to the muscle, the receptor is muscle spindle, the neurotransmitter involved are glutamic acid in the central synapse, For example, monosynaptic reflexes are knee jerk. When a patellar tendon is tapped by patellar hammer, it stretches the quadriceps, and stretched muscle contracts reflexly elevating the leg. Reaction time, it is a time between the application of stimulus and the response. For example, for knee jerk, it is around 19 to 24 milliseconds. So there can be central delay. It is the time taken for the reflex activity to traverse the spinal cord. The central delay for the knee jerk is 0.6 to 0.9 milliseconds. Since the minimum synaptic delay is 0.5 seconds, therefore only one synapse could have been traveled for this reflex activity. Stretch reflexes are thus called monosynaptic reflexes. Coming to the muscle spindle, which are a sensor receptor for stretch reflex. The muscle spindle contains two types of fibers, extrafusal fibers and intrafusal fibers. Extrafusal fibers, they are regular contractile units of the muscle, whereas intrafusal fibers, they are muscle fibers in the muscle spindle, which is the sense organ for the stretch reflex. Each muscle spindle consists of 10 muscle fibers, and these fibers are enclosed in a connective tissue capsule. These fibers are more embryonic in character. The fibers in the spindle have less distinct striation than the rest of the muscle fibers in spindle. The ends of the capsule of the spindle are attached to the tendons at either end of the muscle, or the sides of extrafusal fibers. Only the ends of intrafusal fibers are contractile. There are two types of intrafusal fibers in mammalian muscle spindles. These are called as nuclear bag fibers and nuclear chain fibers. Nuclear bag fibers, they contain many nuclei in a dilated central area, and so they are called as nuclear bag fibers. There are two nuclear bag fibers per muscle spindle, and nuclear bag fibers are of two types. Nuclear bag fiber 1 with low levels of myosin ATPase activity, and nuclear bag fiber 2 with a high level of myosin ATPase activity. Nuclear chain fibers, they are thinner and shorter and lacks definite bag. There are 4 or more nuclear chain fibers per muscle spindle, and their ends connect to the sides of nuclear bag fibers. If you see the sensory innervation to the spindle intrafusal fibers, it occurs through the group 1A and 2 sensory fibers. Sensory nerve endings in each spindle are of two types. They are primary endings and secondary endings. Primary are annulospiral and secondary are flower spray endings. Primary endings are the termination of rapidly conducting group of 1A type of afferent fibers. One branch of IA fibers innervates nuclear bag fiber 1, and another branch innervates the nuclear bag fiber 2 and the nuclear chain fibers. These sensory nerve fibers wrap around the center of the nuclear bag and nuclear chain fibers. And secondary fibers, they are the terminations of group 2 sensory nerve fibers and are located near the end of the intrafusal fibers, but only on the nuclear chain fibers. If you see the motor innervation to the muscle spindle, it is the only organ receiving motor innervation, and the motor innervation comes from gamma efferents, that is of flexor, the smaller motor neurons, and they go to exclusively to muscle spindles. The endings of gamma efferent fibers of two types, plate ending on the nuclear fibers, they are nominated as gamma 1, and trail ending on the chain fibers, they are nomenclated as gamma 2 fibers. Beta motor neurons, they are the larger motor neurons, and they innervate both intrafusal as well as extrafusal fibers. This is the diagram showing the intrafusal fibers of the muscle spindle. They are the nuclear bag fibers, they are nuclear chain fibers. Nuclear bag fibers, they have a dilated portion in the middle. And if you see the innervation, they have a primary and secondary afferents, whereas primary afferents, they come from both the annulospiral endings of nuclear back fibers and chain fibers. Whereas secondary afferents, they are going to arise from the flower spray ending, which comes from the nuclear chain fibers. And if you see the gamma innervation, that is the efferents to the muscle spindle, the gamma 1 is going to innervate both the nuclear chain fibers and back fibers as a plate endings and trail endings, whereas gamma 2 fibers, they innervate mainly uh, the nuclear chain fibers as a trail endings, whereas beta efferent, they innervate both the intrafusal as well as extrafusal fibers. Response of muscle spindle to stretch. Functionally, muscle spindle shows two types of responses, that is dynamic and static. The dynamic response is by nuclear back fibers. Here the primary sensory nerve endings discharge impulses at rapid rate when muscle is rapidly stretched. They fire less rapidly if stretch is continued. The discharge of impulses starts only if there is a change in the degree of stretching of the muscle. Thus, the response depends on the rate of change in the length of the muscle. Whereas static response is seen by nuclear chain fibers. When stretch is continuing, static response comes into play. Thus, muscle spindle gives response to change in the length of the muscles as well as rate of change in the length. Mechanism of activation of muscle spindle. Intrafusal fibers are parallel to the extrafusal fibers. When the extrafusal fibers are passively stretched, the stretch is transmitted to the intrafusal fibers. There will be stimulation of 1A fibers, which causes stimulation of the motor neuron and which leads to contraction of the extrafusal fibers, which constitutes the stretch reflex, and that leads to shortening of the muscle belly. Stimulation of gamma efferent produces the contraction of the intrafusal fibers. The stretches that stretches the nuclear back portion of the spindles, and again there will be stimulation of 1A sensory fibers. Again, that is going to stimulate motor neuron, which causes contraction of the extrafusal fibers. Again, it is going to lead to stretch reflex, causing the shortening of the muscle belly. Thus, muscle can be made to contract via stimulation of a motor neuron that innervate the extrafusal fibers directly, or stimulation of gamma motor neurons that initiate contraction indirectly via stretch reflex. Stretching causes contraction of the extrafusal fibers and shortening of the muscles. Thus, spindle discharge decreases. The spindle and its contraction constitute a feedback circuit, thus maintaining the length of the muscle fiber. Functions of stretch reflex. It is important for maintenance of a posture. Maintaining posture, that is, or changing the posture, are affected by the appropriate adjustment of gamma motor neuron activity of different group of muscles. The role of muscle spindle in voluntary motor activity. Normally, for initiation of skeletal muscle contraction, stimulation of motor neurons are necessary. Along with the motor neuron firing, the gamma motor neuron also fires concomitantly so that skeletal muscle contraction can be made effective, and which is called as alpha gamma linkage. Imagine what would have happened if this do not occur. Stimulation of the motor neuron causes initiation of the contraction of the skeletal muscle fibers, but this contraction will cause, as there is now shortening of the extrafusal muscle fibers, relieving the stretch on the nuclear back fibers, causing the 1A afferents to stop its discharge. This in turn will lead to lengthening of the extrafusal fibers, that is cancellation of the motor stimulation effect. The concomitant firing of gamma efferent is going to prevent this. Other function is damping action. An important function of stretch reflex is its ability to prevent oscillation or jerkiness of the body movements. This effect can also be called as a signal averaging function of muscle spindle. And the fourth function is also play an important role in the maintenance of the muscle tone. Inhibition of stretch reflexes. Because of its monosynaptic pathway, the inhibition of the stretch reflex is produced by pathways that end with the inhibition of alpha motor neurons, and this is brought by two mechanisms, that is by reciprocal innervation and inverse stretch reflex. Reciprocal innervation. When stretch reflex occurs, the muscle that antagonizes the action of the muscle involved relaxes. That is, when agonist contracts, the antagonist is relaxed. This occurs because of the reciprocal innervation. So here in this reflex, receptor is muscle spindle, afferents are 1A fibers, the center is spinal cord, and efferents are alpha motor neuron to the antagonist muscles. In the spinal cord, a collateral branch from 1A fibers arises. It synapses with the inhibitory interneuron, which is called as Golgi bottle neuron, and that synapses directly on motor neuron supplying the antagonist muscle. The pathway mediating this effect is bicenaptic, so because there are two synapses involved. So this is example of postsynaptic inhibition. Significance of reciprocal innervation is that it ensures the movement of the agonist muscle and is not obstructed by the contraction of the antagonist muscle. So here when the muscle is stretched, okay, so it is going to lead to cause contraction of the agonist muscle, and one of the collateral from afferents, it goes to the interneuron, which is going to inhibit the motor neuron of antagonist muscle, and it leads to the relaxation of the antagonist muscle, and this is called as reciprocal innervation. Inverse stretch reflex, or which is also called as autogenic inhibition. As the strength of the stretch increases, the reflex contraction also increases. But after a certain extent, as the strength of the stretch increases, the contraction ends and the muscle relaxes. And this relaxation response to strong stretch is called as inverse stretch reflex. Here, the receptors are Golgi tendon organs, afferents are 1B myelinated nerve fibers, and the center is spinal cord. The 1B fibers end on inhibitory interneurons that in turn end directly on the motor neurons. The efferent shears are alpha motor neurons to the muscle fibers. Golgi tendon organ. So this organ consists of a net-like collection of knobby nerve endings among fascicles of a tendon, which is responsible for autogenic inhibition. There are 2 to 25 muscle fibers per Golgi tendon organ, and stimulation of these 1B fibers leads to the production of IPSP on motor neurons that supply the same muscle from which the fibers arise. Since Golgi tendon organ are in series with the muscle fibers, they are stimulated by both passive stretch and active contraction of the muscles. The threshold of Golgi tendon organ is low. So this is the structure of the Golgi tendon organ, which is present at the end of the muscle fibers. And when the Golgi tendon organs, they are stretched excessively because of the excessive passive stretching or active contraction of the muscle fiber, the afferent 1B fires, and that is going to act on the goes to the interneurons, where it is going to produce inhibitory neurotransmitter on the same motor neuron, which causes the relaxation of the extrafusal muscle, which is called as inverse stretch reflex or autogenic inhibition, which is again example for bicenaptic reflex. Significance of stretch reflex. It is a protective reflex that prevents the tearing of the muscle by extreme stretch. It is important in maintaining tension during normal muscle activity. It is responsible for clasp-knife effect or lengthening reaction. It is a response to a spastic muscle, that is high resting tone, to the stretching or lengthening due to operation of stretch reflex and inverse stretch reflex. When muscles are hypertonic, Moderate stretch produces reflex contraction of the muscle due to stretch reflex, while strong stretch produces muscle relaxation due to the initiation of inverse stretch reflex. For example, if triceps muscles become spastic due to any cause, passive flexion of the elbow meets immediate resistance due to the initiation of the stretch reflex in the triceps muscle. Further, stretch activates the inverse stretch reflex. As a result, the resistance to flexion suddenly collapses and arm flexes. However, continued passive flexion stretches the muscle again and the sequence may be repeated. This sequence of resistance followed by relaxation when a limb is moved passively known as clasp-knife rigidity or clasp-knife effect because of its resemblance to the closing of a pocket knife. It is also known as lengthening reaction because it is a response of a spastic muscles to lengthening. Coming to polysynaptic reflexes or withdrawal reflexes, when more than one synapse is involved in the reflex pathway, it is called as polysynaptic reflex. The reflex path branches in a complex fashion. Because of the difference in the number of synapses, the synaptic delay varies. The activity in the branches with the fewer synapses reaches the motor neuron first, followed by the activity in the longer pathways. This will lead to prolonged response in motor pathways. Some of the branched pathways turn back on themselves, permitting activity to reverberate until it becomes unable to cause prolonged transsynaptic response. Such circuits are called as reverberating circuits. These are common in brain and spinal cord. For example, withdrawal reflex. Withdrawal reflex, it is a typical polysynaptic reflex. Stimulus here occurs in response to noxious and usually painful stimulation of the skin subcutaneous tissue of the muscle, and the response seen is contraction of the flexor and inhibition of extensor. The effect is the part stimulated is flexed and withdrawn from the stimulus, that is, you see flexor response, and if stimulus is still stronger, the response includes flexion and withdrawal of that limb and extension of the opposite limb, and this is called as cross extensor response mechanism. If the strength of the stimulus is more, the excitatory impulse spreads up and down in the spinal cord to more and more motor units, and this phenomena is called as irradiation of the stimulus, and the increase in the total motor units is called as recruitment of motor units. Importance is it's a protective reflex, and the flexion of the stimulated limbs get away from the source of irritation, and the extension of the other limb supports the body. The pattern presumed by all the four limbs puts the animal in a position to run away from the offending stimulus. So the characteristics features of withdrawal reflex. Withdrawal reflexes are prepotent, that is, they prevent the spinal pathways from any other reflex activity taking place at that moment. A weak stimulus can cause quick flexion movement, whereas a strong stimulus causes prolonged flexion and sometimes a series of flexion movements. The prolonged response is due to the prolonged repeated firing of motor neurons. The repeated firing is called as after discharge, which is due to the continued bombardment of the motor neurons by impulses arriving by complicated and circuitous polysynaptic, that is, reverberating circuits. So this is the diagram of reverberating circuits because of the involvement of the mini synapses, which is supposed to carry impulses to the motor neuron, and sometimes some of the synaptic activity goes back to the previous neurons, and they causes the reverberating circuits, and which is going to lead to the after discharge, which leads to the prolonged effect to the motor neuron, which is called as after discharge. Local sign. It refers to the ability of reflex to confine the withdrawal exactly to the portion of the body affected by noxious stimulus. For example, if the medial surface of the limb is stimulated, the response will include some abduction with flexion, whereas stimulation of the lateral surface will produce some adduction with flexion. This dependence of exact response to the location of the stimulus is called as local sign. Some of the general properties of reflexes are they require adequate stimulus. The stimulus that produces reflex is very precise as the receptors respond maximally only when an appropriate specific stimulus is applied. It is called as adequate stimulus for the particular reflex. Final common pathway. The alpha motor neurons that innervate the extrafusal muscles are the final common pathway over which the neural control system coordinates the activity of skeletal muscle fibers. It serves both as an integrating center and efferent pathway. Central excitatory and inhibitory status. The spinal cord shows prolonged changes in the excitability because of activity in reverberating circuits or prolonged effects of synaptic mediators. The prolonged state in which the excitatory influence exceeds inhibitory influence is called central excitatory state, and conversely, it is inhibitory state dominates, it is called as central inhibitory state. For example, after complete transaction of the spinal cord, when reflex movements return, a mild noxious stimulus on the lower limb may cause withdrawal reflex, and autonomic changes like urination, defecation, sweating, and fluctuation in the blood pressure, which is referred to as mass reflex, which occurs because of the central excitatory state in the spinal cord. Habituation and sensitization of reflex responses. Though the reflexes are fixed, they can still be modified by experience. If the stimulus is benign and repeated at frequent intervals, the response declines and disappears. This phenomena is called as habituation. It is due to the synaptic fatigue, which is due to the exhaustion of the chemical transmitter, as its synthesis is not rapid as its release. The decrease in the release of neurotransmitter can also occur due to the gradual inactivation of calcium channels at presynaptic terminal. This decreases the intracellular calcium and the release of neurotransmitter. So this is in brief about reflexes, the different classification of reflexes and properties of reflexes. Thank you.